week's Neet discussion. So topic granular, that is photosynthesis. Right? First question: conversion of pyruvate into phosphoenol pyruvic acid takes place. So this is a C4 cycle. Okay. In C4 cycle, the primary carbon dioxide acceptor is 3-carbon compound, that is phosphoenol pyruvic acid. This phosphoenol pyruvic acid will fix the carbon dioxide in presence of enzyme, that is PEP carboxylase enzyme, phosphoenol pyruvic carboxylase enzyme. You have PEP carboxylase enzyme, and you will see the first stable compound formed, that is oxaloacetic acid. Oxaloacetic acid. This is a 4-carbon compound, first stable compound. That's why this cycle is known as C4 cycle or C4 pathway. So conversion of pyruvate into phosphoenol pyruvate takes place in. See, in case of C4 plants, there are two types of cells in which carboxylation occurs. The two types of cells. One is mesophyll cells, which are surrounding the vascular bundle, outside the vascular bundle. Okay. Then another one, that is bundle sheath cell. Bundle sheath cells. Bundle sheath cells are parenchymatous cells, which are immediately surrounding the vascular tissue. Okay. So mesophyll cells, normal chloroplast, smaller size, having both grana and stroma. Bundle sheath chloroplast are having only stroma, agranal chloroplast. Right? So this method of carbon dioxide fixation, first it occurs in mesophyll cells. Then in bundle sheath cells, C3 fixation. Okay. The carbon dioxide for C3 cycle will come from the decarboxylation of the malic acid. Malic acid, when it undergoes decarboxylation, it releases carbon dioxide. That carbon dioxide is accepted by RBP. Then Kelvin cycle occurs in what you call the bundle sheath chloroplast. And this reaction mainly occurs in the chloroplast or mesophyll cells. So answer will be mesophyll cell chloroplast. So 1, 2. Next, which one is correct for C4 plants? See the question here properly. Mesophyll, bundle sheath, one. Mesophyll, first option, PEP case, C4 cycle, Rubisco, C3 cycle. So we will go with the first option. First option is correctly matched here. Is it or not? So this phosphoenol pyruvic carboxylase enzyme, in short, we will write it as peptide enzyme. Okay, it is involved in C4 fixation. In case of C3 pathway, you have ribulose 1,5 biphosphate fixed carbon dioxide. Ribulose 1,5 biphosphate fixed carbon dioxide in presence of which enzyme? Rubisco enzyme, and it results in the formation of a first stable compound, usually that is 3-PGA, a 3-carbon compound, phosphoglyceric acid. Okay, so Rubisco catalyzes the carboxylation in C3 plant. Okay, here ribulose 1,5 biphosphate is acceptor of carbon dioxide, and in C4 plant carbon dioxide is fixed with phosphoenol pyruvic acid. So second question, first choice is correctly matched. Next now, photosystem 1 is located on. So photosystem 1, or you have pigment system 1, whatever you call. Based on the order of their discovery, you have photosystem 1 and photosystem 2. This is the double membrane bound cell organelle. That cell organelle is chloroplast. Okay, so you have like this are there. This part, what you are seeing, this is non-appressive. Unappressive. Non-appressive or unappressive, whatever you write. And these regions on both sides, these are sandwich appressive. This is non-appressive. This is non-appressive part of thylakoid membrane. This is connected to another granum. Thylakoid stacked one above the other, resulting in the formation of granum. This you call it as stroma lamellae or red channels. Okay, so PS1 is situated in non-appressive membrane, non-appressive region of thylakoid membrane, and it is present here in stroma lamellae or red channel or intergrana lamellae. So here answer will be, what is that? Both 1 and 2. Okay, non-appressive part of grana thylakoid, and this is stroma thylakoid, that is intergrana lamellae, this one. So both 1 and 2. So third option, this third question, 4 is the answer. Next, in cycle, if one molecule of RUBP is carboxylated, then how many PJ molecules will be formed? So here, Kelvin cycle they are asking, RUBP, how many molecules? One molecule they are asking. It is point carbon compound. It is fixing carbon dioxide in presence of Rhodesico enzyme. So first stable compound, it is 3-phosphoglyceric acid. It is 3 carbon compound we know. So 6 carbon compound, this 5, this 1, total 6 carbon. And it is 3 carbon compound. Here one molecule, 3 carbon compound means it should be 2 molecules. Total 6 molecules of carbon dioxide are fixing 6 molecules of carbon dioxide. RUBP is being fixed by RUBP fixes 6 carbon dioxide. Okay. So when RUBP fixes 6 molecules of carbon dioxide, then how many molecules of the PJ you are going to get? 12 molecules of 3 PJ. So here, what can be answered? That is 2. Okay. So 4. Next. Malic acid or aspartic acid and oxaloacetic acid are both found in say this is the chloroplast of mesophyll cell. This is the chloroplast of bundle sheath cell. So initial primary carbon dioxide fixation by phosphoenol pyruvic acid, okay, in presence of peptidase enzyme, it results in the formation of 4 carbon compound, that is oxaloacetic acid. Oxaloacetic acid can be reduced into malic acid, or even it can be reduced into aspartic acid. Aspartic acid. So here There is utilization of reducing power NADPH2, and what it produces NADP plus. Okay, so what is got to malic acid will get due to reduction process, or you can get even aspartic acid also. Okay, so this is catalyzed by malic or malate dehydrogenase enzyme. Okay, so oxaloacetic acid, malic acid, or aspartic acid are found in where they are found in the mesophyll cell. They are found in the mesophyll cell. So fifth, it will be first option. 5, 1. Next, biosynthetic phase of photosynthesis is dependent on. See, biosynthetic phase means it is a dark reaction. It is a dark reaction, or it is known as a biochemical reaction. It is usually most common method of carbon dioxide fixation during dark reactions, that is Kelvin cycle. Okay. These occur in the stroma region of the chloroplast. So, for the occurrence of these reactions in the stroma, they need the product of light reactions, photochemical reaction, light reaction. Without these products, biochemical reaction or dark reactions will not proceed. Okay, so we know light reaction products. The light reaction products mainly oxygen is liberated as byproduct. Okay, then we get ATP, energy currency. ATP are produced by this photophosphorylation, cyclic or non-cyclic, mainly non-cyclic. A synthesis of ATP is explained by chemiosmotic hypothesis as given by Peter Mitchell. Okay. Then one more you have NADPH2, that is strong reducing power. Strong reducing power. So these are assimilatory powers. So these are used in Kelvin cycle for carbon dioxide fixation. So here, biosynthetic phase of photosynthesis is dependent on NADPH2 and ATP. So it is first option. So 6, 1. NADH2 is not used. NADH2 that comes in respiration process, not in photosynthesis. Next, Kelvin cycle is also called. See, it is known by different names as I have given. It is a dark reaction, biochemical reaction, along with Kelvin, his friends, his co-workers assisted him in this research work. They did research on chlorella and even Scannidus using radioactive level carbon dioxide, that is C14. Radioactive level carbon dioxide, that is C14. Okay, so they found out, they traced a complete chemical pathway of carbon dioxide fixation to find out how the carbohydrates are produced during the dark reactions independent of light by using these assimilated powers. So here you have Andrew Benson, and James Pashan. Andrew Benson and James Pashan, they assisted the Kelvin in finding out this complete set of chemical reactions. That is why, in the memory of them, it is called as the Kelvin-Benson cycle or Kelvin-Benson-Pashan cycle. Okay, so different names are given to it. And another name is there, that is RBB. So here, The 5-carbon keto sugar, RUBP, RUBP is 5-carbon keto sugar. This will be reduced into glucose, carbohydrates. That's why it is also known as reactive pentose transit. Yeah. So here, seventh, we will go with all of these. We go with all of these. So seventh, it is 4 terms. Next now, 8th one. Promisco is found in, see, it is one of the most abundant enzyme protein present in the chloroplast. 16% of the protein in chloroplast is constituted by Promisco. So Promisco is found in chloroplast, which is catalyzing carboxylation step in C3 cycle. So it is 8-4. See, in mitochondria, we have respiratory enzymes. In mitochondria, respiratory enzymes will be there. In chloroplast, photosynthetic dark reaction enzymes will be there mainly. Next, in dark cycle, that is C3 plants, one molecule of glucose formation needed. See, one molecule of glucose. We learned the Kelvin cycle, right? In Kelvin cycle, to fix 6 carbon dioxide, number of ATP used 18 ATP. After the fixation of 6 carbon dioxide, there will be synthesis of one molecule of glucose. And how many NADPH are produced? 12 NADPH are used to fix 6 molecule of carbon dioxide. Okay. Now in this question they are asking, okay, for one molecule of glucose formation, how many ATP and NADPH are required? They are asking. So it is 4th option. It is 4th option. So 9 over. Now they may ask us to fix one carbon dioxide, how many ATP and NADPH are required? Then it is 3 ATP and 2 NADPH. So this you have to remember in the case of C3 pathway. Suppose if it is C4 pathway, then number is different, you know that 30 ATP and 12 NADPH2 for fixing 6 molecule of carbon dioxide by C4 pathway. So in C4 pathway, to fix one carbon dioxide, so there they use 5 ATP and 2 NADPH. So this is in C4 pathway for fixing 6 carbon dioxide. These 12 additional ATP will be used because conversion of AMP to ATP, there it makes use of 2 ATP. So 6 into 2, 12 additional ATPs are required. Normally in C3 cycle 18, here 12 extra. So it comes to 30. Next. If the light becomes unavailable during photosynthesis, see, when light is there, usually you can see operation of photochemical reactions, like reactions, real reactions. So these can result in the synthesis of ATP and NADPH2 with the liberation of oxygen due to splitting the water. So what they're asking, if the light becomes unavailable during photosynthesis, means if light is not available, then it can be done. So Usually in that which reaction occur? Biochemical reaction, or what we say that reaction, Kelvin cycle occurs. Okay. How long the concentration or molecules of these are there, carbon dioxide fixation will continue. Once all these ATP and NADPH2 are used in this C3 cycle, further there is no continuation of that reaction or C3 cycle. Once ATP and NADPH2 get exhausted, these are required for C3 cycle. Okay, so answer can be biosynthetic phase continues for some time and then stops. When it stops, when all the molecules of ATP and NADPH are used up, no more these molecules are available, then the dark reaction can cease or stop. Okay, once again, if you provide light, definitely light reaction occurs, synthesize ATP and NADPH2, then dark reaction can continue independent of light. Okay, so 10th one is fourth. Next, which of the following statement is false in case of C4 plant? False statement they are asking. One by one we'll read. Carbon dioxide acceptor is visco in mesophyll cell. First statement itself is wrong. That is because in C4 cycle, carbon dioxide acceptor is PEP, phosphoenol pyruvic acid. In presence of which enzyme? Peptidase enzyme, phosphoenol pyruvic carboxylase enzyme. So peptidase enzyme catalyze carboxylation in C4 plant, not Robisco. So that is why first statement is wrong. Carboxylation occur in mesophyll cell, correct? Okay. The initial carbon dioxide fixation occur in mesophyll cell only, that result in 4-carbon compound formation. Then leaves have two cell types: leaves, bundle sheath cells and mesophyll cells, right? Then mesophyll cell lack Robisco, right? Mesophyll cells do not have Kelvin cycle enzyme. Similarly, bundle sheath chloroplast do not have C4 cycle enzymes. That carboxylase enzyme will not be there, right? So 11th one will be carboxylation is the fixation of carbon dioxide into. So carboxylation, when it occurs, this carboxylation can result in the synthesis of carbohydrate. Carbohydrate is organic compound. Series of steps will be there, not a simple reaction. Series of chemical reactions which will be occurring that finally result in the formation of sugar, that is carbohydrate, organic compound. So answer, it can be fourth option, organic intermediate. Directly you will not get sugar. In between 3-PGA, 3-PGAL, dihydroxyacetone 3-phosphate, so different intermediate organic compounds are formed. Okay, these 3-PGAL and DHAP, they take part in what we call sugar formation on them. So it is the 12th one is fourth option. Okay, next now, PEP is present in phosphoenol pyruvic acid. So PEP is present in mesophyll cell. Chloroplast of mesophyll cells. Chloroplast of mesophyll cells, they have PEP, phosphoenol pyruvic acid. Next, PEP has an advantage over Robisco. The advantage is, so 13(1). So moving on to the protein, the phosphoenol pyruvic carboxylase enzyme has an advantage over Robisco enzyme. See, in C3 plants, carboxylation is catalyzed by Robisco. Okay, this enzyme has receptor sites where either oxygen or carbon dioxide can bind. I told which gas should bind to this enzyme. That is determined by relative concentration of carbon dioxide or oxygen. Okay, suppose If CO2 level is more, then Rubisco catalyzes carbon dioxide fixation. So it acts as RUBP carboxylase enzyme. Suppose if the relative concentration of oxygen is more, then it will start fixing oxygen instead of fixing carbon dioxide. Then it can act as RUBP oxygenase. So when the regulose biphosphate carboxylase enzyme, when it starts fixing oxygen, then there is a process called photorespiration. Because of this photorespiration, photosynthetic yield will be reduced. So C3 plants can perform photorespiration because of the dual nature of rubisco, whether it's binding with carbon dioxide or binding with oxygen. Whereas this PEP carboxylase enzyme, PEPase enzyme, irrespective of the concentration of the carbon dioxide in the surrounding air, it show usually affinity towards carbon dioxide only, not towards oxygen. That's why C4 plants do not perform photorespiration, right? So answer will be Rubisco combined with oxygen, but that case do not combine with oxygen. So here it is the 14th one. It is first option. 15th you see now. Bundle sheet cells are rich in which enzyme they are asking? Listen, bundle sheet cells. So in C4 plants you have mesophyll cells and bundle sheet cells. So in C4 plants, bundle sheet cells or chloroplast cells, they have Rubisco enzyme. Is it or not? They have Rubisco enzyme. Now, PEP carboxylase, PEPase, where it is presented in the chloroplast of mesophyll cells. The chloroplast of the mesophyll cells. Okay. Malate dehydrogenase enzyme. This we learned in C4 pathway only. Okay. Oxaloacetic acid, which is formed in the first step after carboxylation, it undergoes reduction using a NADPH2. Okay. And what you will get? Malic acid. Under 4 carbon compound. Right? This reaction is catalyzed by the malate dehydrogenase enzyme or malic dehydrogenase enzyme. Then third, they have given fructokinase. This, in case of glycolysis, respiration step. Okay. You have glucose to glucose 6 phosphate. Glucose 6 phosphate to fructose 6 phosphate. This is step number 1, where ATP can be used. This is step number 2, where ATP is used. Okay, used. This is isomerization, glucose 6 phosphate to fructose 6 phosphate. We will learn this in respiration term. Now, this third step, fructose 6 phosphate to fructose 1,6 diphosphate you have. Okay, where ATP is used once again. ATP is used. This step is catalyzed by this enzyme, that is phosphofructokinase. Catalyzed by phosphofructokinase enzyme. So here, 15th, what is that answer? 4th, then 16th. Now, carbon dioxide released in bundle sheet is used in decarboxylation of malic acid result in carbon dioxide release. That carbon dioxide is used in C3 cycle for the sugar synthesis. So it is 16. What it is? 2. 17. Every carbon dioxide molecule entering the Kelvin cycle needs. Every carbon dioxide molecule entering the Kelvin cycle, what it requires? Molecules of NADPH2 and 3 molecules of ATP for carbon dioxide fixation, right? So 17 will be 1. Next, 18. Primary acceptor of carbon dioxide in C4 cycle is phosphoenol pyruvic acid. Okay, so it is 2. 18 2. PGA is the first stable compound in C3 plants. Oxaloacetic acid is the first stable compound in C4 plants. Okay, listen. They have given characters here. They have special leaf anatomy. It is usually called as crank anatomy. They tolerate high temperature and lack photorespiration. They don't have perform photorespiration, and photosynthetic productivity yield is very high. Okay, greater productivity of biomass, organic matter. So these are all the characters of which plants? C4 plants, right? So 19 will be 3. In non-reaction, C3 plants, regeneration of RUBP needs 1 molecule of RUBP formation, regeneration 1 ATP. Is it or not? For 6 RUBP molecule regeneration, total 6 ATP will be used, right? So your 20th will be first option. In C4 pathway, the first product identified was oxaloacetic acid. So it is 21. 2. Next, 22. The electron in the reaction center of PS1R. So the electron in the reaction center of PS1R. So here, during that scheme, or non-cyclic photophosphorylation, PS2, P680, you have electron acceptor. Here you have ETS. Okay, ATP synthesis. Then here what you have? PS1, P700. The light electron acceptor. Then it finally moves to NADP plus and it gets reduced to NADPH2. Here splitting the water molecule into there. So this graph, what we call representation of non-cyclic phosphorylation or Z scheme. So here, what they are asking, the electrons in the reaction center of PS1 are excited simultaneously. Both can receive the light simultaneously. This is absorbing 680 nanometer red light. This is absorbing 700 nanometer red light. So answer will be either 1 or 2. See here, excited simultaneously with PS2 or excited simultaneously with P680. That is the reaction center in the photosystem 2. So it is 4th option, 22-4. Next, 23. Within the chloroplast, the chlorophyll pigments are organized in the form of pigment system. We have 2 pigment system or photosystem. Photosystem 1 and photosystem 2. This photosystem contain reaction center. They contain electron carriers. Okay, they contain light harvesting complex. These light harvesting complex are made up of different kind of pigments, chlorophylls, carotene, and xanthophyll. Reaction center mainly chlorophyll A molecule, either 680 or 700. So answer will be 23rd, 4th. That is both 1 and 2. Chlorophyll pigments are organized either in the form of photosystem 1 or photosystem 2. 24th. In photosystem 2, the reaction center chlorophyll A absorbed. So chlorophyll A here, chlorophyll A absorbed 680 nanometer wavelength of light, red light, causing electron to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of here it contains cytochromes, iron containing pigment protein which act as electron carrier. So answer will be third option. 24 third. Okay, lastly, the water splitting complex is associated with photosystem 2. It has oxygen evolving complex. This oxygen evolving complex has 4 manganese ion and chloride ion. It is concerned with the splitting of water molecule. Okay, photolysis of water. Oxygen evolving complex associated with photosystem 2. So answer is second. Right? So these are the questions of today's discussion paper. Right? So tomorrow on this basis you will write the weekly test. Theory syllabus I will continue as usual on Tuesday.
Because it's also need exam. Need test. Topic is products in this elimination. Now, the question number 1. This is the zoology part. The level of metabolic waste in the form of urea is called ureotenism. So as you know that the most of the organisms in a different aquatic medium, they are eliminating the different nitrogenous waste materials, which are in the form of either it is urea or in the form of the uric acid or it is in the form of ammonia or it is in the form of amino acids. Okay. The animals which are eliminating the urea as a nitrogenous waste material, these are referred as ureogenic animals. The phenomena is called as ureotenism. The animals which are eliminating the nitrogenous waste material in the form of uric acid, they are referred as uricotenic animals. The phenomena is called as uricotenism. The animals which are eliminating the ammonia as a nitrogenous waste material, those are referred as ammonotenic. The phenomenon is called as ammotelism. The animals which are eliminating the amino acids as a nitrogen waste material, these are referred as aminogenic animals. The phenomena is called as aminogenism. Okay. So where the urea is produced during detoxification of amino acids in liver. It is produced during detoxification of the amino acids. So this all these processes, that means the nitrogen waste material elimination or maybe the formation of these end products is mainly occurs in the liver. Okay, because the most of the nutrients, that is most of the protein synthesis is occurs in the liver, even though waste material is also protein. Okay, so here this urea is produced during the detoxification of the amino acids. At the same time, this uric acid is produced due to the breakdown of the nucleic acids. Nucleic acid metabolism, which induces one of the end product, is called as uric acid. So such type of uric acid is eliminated by an individuals in the form of the semi-gel. Okay, so this is commonly observed as in case of the reptilians, as in case of the birds. Okay, similar. But whatever the reptilians are eliminating, these are not some of the reptilians, not all the reptilians, a few of the reptilians, and most of the birds, they are eliminating the uric acid as a nitrogen-based material. Okay, which is excreted in the form of the semi-jelly-like structure. Okay, likewise, ammonia is excreted due to a process is called as this is the ammonia is produced as one of the end product during the deamination of amino acids. Deamination of amino acids. This ammonia is released as one of the end products. Okay. That's why those individuals are referred as aminogenic animals. Okay. So example for this one is almost all the fishes, they are eliminating the ammonia as a nitrogen-based material. Okay. Amino acids are the excretory products which are eliminated during the deamination process. At the same time, whatever the amino acids are produced as a end product, or it might be due to the metabolic activity, whatever the end products are produced in the form of amino acids, those are eliminated as a nitrogen-based material. And this is common as in case of some of the individuals like the spider. The spider is going to eliminate one of the amino acid as a nitrogen-based material. It is called as guanylic acid. The guanylic acid is one of the amino acid. This occurs during the metabolism of nucleic acid. It is produced as one of the end products, nitrogen-based material. So that will be eliminated by these spiders. Okay. So that's why your option is the second one. Next, the uric acid is produced by the breakdown of the uric acid is produced by the breakdown of the amino acids, as I said here. Okay. Next, the starch is commonly called as glycans. It's called as glycans. So such type of starch is occurs in the form of the starch grains in the seeds of the plants. The plants is mainly possessing, they store this starch content in the form of the starch seeds. That is aileron materials will be there. Aileron material is the occurs in the form of raphides. These are occurs in the form of extrinsic starch. These are occurs in the form of intrinsic starch. Okay. So such type of starch, they are commonly called as glycans. These glycans are, it might be the homoglycans or it is heteroglycans. Homoglycans means the starch polymer is made up of only one type of sugar molecule. If it is one of the polysaccharide chain, if it is made up of only glucose monomer unit, more than 10 glucose monomer units, if they are undergo condensation process and it produces one type of polysaccharide chain that is called as same type. It's made up of the homoglycans because it is made up of only similar type of sugar monomers. Suppose if it is made up of the different sugar monomers. Suppose it is made up of glucose plus galactose plus glucose plus fructose, like this. The many monomer units, they are undergo the polymerization process, condensation process, by which it produces one of the polysaccharide chain. Such type of polysaccharide chain are referred as heteropolysaccharides. Okay. Next, the guano, the fecal matter of birds contains insoluble crystals of uric acid, even though insoluble crystals of urates also. Insoluble crystals of uric acid and insoluble crystals of urates. Okay, so that's why both 1 and 2. The flame cells are excretory structures of the flame cells are the excretory structures of the flatworms. That is, platyhelminthes individuals, especially the tinea solium, is mainly possessing the one of the excretory structure is called as the flame cells. This flame cells is occurs like this. Okay. Inside the cells, the filament-like structure, they are arranged in the form of the flame-like structure. So that's why they are referred as the flame cells. These are the excretory structures of the platyhelminthes individuals, especially the tinea solium and other organisms belongs to this platyhelminthes. So these are also called as solenocyte cells. These are also referred as solenocyte cells. The annelids are mainly possessing metanephridia are they as a excretory structures. Metanephridia. Even the some of the annelids like the earthworm not only possessing the metanephridia, even though they are also possessing some other type of structures which are involving in excretion. Okay. Even the annelids like the leech, also some other structures are there. The leech botryal tissue is there, which is serves as excretory function. Botryal tissue is serves as excretory function. This is as in case of leech. Okay, so that's why here the question number 4 option is the third one. Arthropods, they are possessing Malpighian tubules, not Malpighian body. Malpighian tubules are different, Malpighian body are different. Okay, arthropods, they are possessing antennal glands, coxa glands, green glands, zippler. Peptone glands are also there, even though zippler. So these are different glands are there, where they are serves as an excretory function. The crustaceans, they are mainly possessing the green gland as a excretory structures. Okay, next. Fifth one. Antennary or green glands, which are excretory and osmoregulatory organs, the crustaceans consist of. They mainly consist of end sac. They mainly possessing end sac. Okay, the end sacs are nothing but these are the convoluted ducts. These are convoluted ducts present in the kidney of such type of individuals. Okay, so that's why these are referred as the excretory structures of crustaceans. But the renal sac is nothing but it is a fluid-filled cysts arrangement in the depressions are there in the kidney part. So the fluid-filled depressions are called as a renal sac. Okay. Likewise, antenna. Antenna is one of the structure. It is a sensory structure where earlier it was believed that it is tactile in function, but actually it is not a tactile. It is olfactory. Okay. Earlier it was believed that this antenna are tactile in function. Tactile means sense of touch. Tactile means sense of touch. But actually these are not tactile receptors. These are olfactory receptors. Next. The kidney not only remove the waste from the blood but also play a very important role in maintaining temperature of the body, equilibrium of the body. So third segment you can see constant composition of the blood irrespective of the nature of the food as well as fluid intake. If the blood osmolality is maintained, automatically the entire body osmolality will be maintained. Okay. So that's why compared to the first two, this is most appropriate ones. Okay. Yes, sir. Like it's here. Blood is playing a vital role in the circulation of these mineral salts, various other substances. The circulating blood is also playing a vital role in maintaining the temperature. Okay. So this is why compared to those, the second option, this third one is most appropriate. Okay. Next. The outer cortex of the kidney in a case of appears granular or dotted because of granular cytoplasm is given. Granular cytoplasm content we are going to observe as in case of the leukocyte cells. PMNL. What do you mean by PMNL? Polymorphonuclear leukocyte cells. This polymorphonuclear leukocytes means whatever the cell possessing the nucleus, this nucleus is mainly modified into the lobed ones. Like this lobed nucleus we are going to observe. This instead of single nucleus, they are possessing lobed nucleus. Like this. This is mainly because of the deposition of the excess of the pigmented granules within the cell cytoplasm. So that's why, so these cells are referred as granular leukocyte cells. Okay. The granular leukocyte cells, best example it is the neutrophils. This one is eosinophils. The basophils are trilobed nucleus. They are possessing trilobed nucleus. Like this. Okay. These are basophils. So all these cells are referred as granular leukocyte cells. These are leukocyte cells. But some of the other cells are also called, some of the other leukocytes, they are called as agranular leukocytes. Agranular leukocytes like the monocytes and lymphocytes. The monocyte cells, they are modified into macrophages once they are comes out from the bone marrow. When they are leave the bone marrow and enter into the connective tissue, macrophages main function is involving in the phagocytic. They are involved in phagocytic function by the secretion of the opsonine content. By the secretion of opsonine. Likewise, so here the lymphocyte cells are the other type of granular leukocytes. So these lymphocyte cells are involved. Humoral immunity as well as cell-mediated immunity. They play a vital role in humoral immunity as well as cell-mediated immunity. Humoral immunity means the antibody-mediated immunity is called as humoral immunity. Cell-mediated immunity is playing a vital role in the graft resection of the transplanted organ. Okay. So that's why. So these are the different types of cells, leukocyte cells. But these are the different types of leukocyte cells. But their function is totally different. Okay. Is it clear? Now, Next, second statement is the presence of loop of health. The presence of loop of health is mainly helps in maintaining and regulating the water concentration as well as solid concentration within the body. Okay. Next, the presence of collecting ducts. The collecting ducts are mainly helps in the concentration of the urine. Then, fourth one, the much convoluted uniferous tubules and Malpighian corpuscles in this region. So, if you take the TS of the cortex region of the testis, sorry, the kidney, then we are going to observe many the Malpighian corpuscles as well as the uniferous tubules we are going to observe. Uniferous tubules like the PCT, BCT, okay, and collecting duct we are going to observe. Apart from that, Malpighian corpuscle. Malpighian corpuscle means the Bowman's capsule along with the granular capillary. It's called as Malpighian body or Malpighian corpuscle. Okay, so that's why option 4 one is correct. The concavity on the medial side of kidney is known as, it's called as hilum. Through this hilum only, the artery will enter into the kidney. Which artery? Renal artery will enter into the kidney. Through this part only, the renal vein comes out from the kidney. Through this part only, the ureter comes out from the kidney, and through this, the nerves will enter into the kidney part. Okay, that is called the hilum or hilus. Okay, next, the renal pelvis is there. Once this hilum is enter into the inner part, kidney part, it is modified into a depressed irregular shape, and that is called as pelvis. In this renal pelvis, there is a storage of urine will be there. Final filtrated urine is gets stored temporarily. At the same time, this pelvis introduces to the 2 to 3 calyces. So these calyces are called as major calyces. Each major calyces which introduces to minimum 6 minor calyces. Okay, that means total number of calyces in each kidney might be this varies from 8 to 18 in number. 8 to 18 in number. Okay, minor calyces number. Then the podocytes are associated with. These are mainly associated with the Bowman's capsule. The Bowman's capsule, if you observe the Bowman's capsule, the parietal layer as well as visceral layer is there. It is made up of 2 layers, parietal as well as visceral layer. So it is like this. So these are called as podocytes. These are modified from squamous epithelial cells. The squamous epithelium, they are modified into the podocyte cells. Next, PCT part of nephron. PCT part of nephron, they are mainly lined by like simple cuboidal epithelial cells, single layer with microvilli. With microvilli. Then, glomerulus is mainly lined by the squamous epithelial cells. Granular is lined by the squamous epithelium. It is also lined by flat squamous epithelial cells. Next, the smallest function unit of kidney is nephron. So, in each of 1 million of nephron units we are going to observe. 1 million of nephrons we are going to observe in each kidney. Next, the endless loop are found in those animals which exhibit hypertonic urine. One of the following does not have endless loop. Which one? Frog. Why? Because the frog has mesonephric kidney. Frog has mesonephric kidney. The types of kidney are: first one is archinephric kidney. Second one is protonephric kidney. Third one is mesonephric kidney. Fourth one is metanephric kidney. Archinephric kidney we are going to observe in the ancestor individuals, especially the extinct ostracodermy. Okay, ostracodermy individuals possessing such type of nephron is called as archinephric kidney. The protonephric kidney, these are smaller ones. These are simple tubular straight tube. Granular capillary is there within the kidney. Those are referred as protonephric. Mesonephric kidney tubules are, nephron tubules are there, but the endless loop is not there. Okay, it's not highly. It is highly convoluted, but the endless loop is not there. It is totally absent. But metanephric kidney is characterized by the presence of endless loop. Okay, then. The thin segment of a descending limb of a lobe of hell is lined by flat cells. It is squamous epithelial cells. The collecting tubes or duct of Pillay. The collecting tubes towards the last end, they are gets widened and they are gets joined to opens into the renal papillae. It is called as the duct of Pillay. The Pitter's canal. The Pitter's canal is observed as a case of frog. Frog kidney possessing one of the duct, where vasa deferens is opens into this duct. This duct is called as Pitter's canal. Okay, is it clear? It's called as Pitter's canal. The frog's kidney possessing such type of canal is called as Pitter's canal. Then, freshwater surface can be taken as a characteristic feature of OCT. OCT is organic cation transport. OCT is organic cation transport. Next, the first fourth, fifth portion of the descending limb is lined by the cuboidal epithelium, simple cuboidal epithelium. Which of the following statement is not true with respect to the nephron? Incorrect statement. Cortical nephrons are more common. Yes, common. Second one is cortical nephrons lack vasa recta. Yes. Third one is in juxtamedullary nephrons, the blood first passes through the vasa recta and then through the capillary of the glomerulus. No wrong. Next. Third one is incorrect statement. Next, 70. Which of the following is correct? Okay. Option third one. Efferent arteriole is narrower than that of apparent arteriole. 18. Retroperitoneal kidney uncovered by peritoneum on its dorsal surface, dorsal end side is called as retroperitoneal kidney. Find out the incorrect statement. Which is incorrect? The kidney contains millions of nephrons. Whether kidney contains millions of nephrons? Only 1 million is there. Each kidney possessing 1 million. Millions of nephrons is given. Where what? Sorry, neurons is there. This is wrong. The third one is wrong. I thought it is nephron. The neuron is there. This is wrong statement. Next, 20th. The opening of the urinary bladder is guarded by two urethral sphincter. Which one is the involuntary function? Internal sphincter is made up of smooth muscles. Smooth muscles are involuntary function. Okay, that's why option first one. Find out the incorrect statement with respect to the cortical nephrons. Cortical nephrons, whether they control the volume of the plasma under stress condition? No. The stress condition, the volume of the blood is controlled by the adrenaline as well as non-adrenaline hormones secreted by the kidney. Okay. Next. So that's why fourth option is incorrect statement. 22nd. Where do you find podocyte cells in human body? Kidney. Proximal colony tubule is highly specialized for reabsorption of substances. It is lined by simple cuboidal epithelium without microvilli. Next. Which of the following cannot be considered as part of structure of uniferous tubule? Collecting duct is not considered as a part of structure of uniferous tubule. It's separate duct it is. Next, 25. The most advanced kidneys in which loop of Henle is present are metanephric kidneys. These are found in all except amphibians. Amphibians possessing mesonephric. Next, last one. Option 8 is first one. 26. Which of the following structure helps in extraction and conservation of water in terrestrial arthropods? It is non-fission liquids. Cables organs are also called as Bojanus organs, observed in molluscs. Okay, it serves as excretory function.